We will be discussing now citrus fruit rot and gummosis. This is the occurrence of the disease. The disease is found in Spain, France, Italy, all Mediterranean countries, USA, Cuba, Paraguay, Brazil, Mexico, etc. In India, it was recorded in the year 1927, that is citrus gummosis on sweet orange. Different states of India like Assam, Maharashtra, Karnataka and Andhra Pradesh were recorded for occurrence of this disease. About economic importance, in Sicily, all citrus trees were destroyed by this disease between the year 1863 to 1870. It is the major cause of citrus decline in Indian conditions. Symptoms. Symptoms of the disease, we can divide it in different types which occur in root, which occurs in different forms like fruit rot, gummosis, leaf fall and fruit rot, and nursery infection. Now we will discuss all these. And root rot, injury and destruction of small fibrous feeder roots by Phytophthora, is revealed by few numbers of rootlets as compared with uninfected root systems. In severe cases, infection progress on main lateral roots through feeder roots and develops bird eye spots on big roots to cause complete rotting of relatively small roots. For foot rot or stump rot, root near the collar regions get infected. Infection often extends to the bark on the trunk. In case of complete girdling of the trunk, plant may be killed suddenly. Now we will discuss about gummosis. Dead areas of bark are produced on a stem that remain firm, and ooze comes out from these areas. Gum oozing is more common in kagsy lime, seedless lime, and in rough lemon rootstocks. In leaf fall and fruit rot, leaf blight symptoms are seen in nursery. Leaf usually drops off before the complete leaf is blighted. All leaves and fruits drop off due to infection. In nurseries, root stalks are suffered by the disease. Phytophthora causes loss of rootstock seedlings in nursery due to pre and post emergence damping off. Unlike Rhizoctonia collar rot, in Phytophthora infections, young seedlings are infected at the growing tip and seedlings fail to grow further. These are the symptoms of citrus gummosis, gum or ooze coming out from the infected part of plant. This is about causal organism. So there are many species of Phytophthora like Phytophthora palmivora, Phytophthora citrophthora, Phytophthora nicotiana parasitica, which are associated with the etiology of this disease. This is disease cycle. Pathogen survives in soil by forming chlamydospores and oospores. If moisture is available, zoospores will be liberated and they will infect the host. When they will reach the host surface, they will encyst, germinate, and cause infection. Dissemination of infective propagules of the pathogen will be by rainwater, irrigation water, or by rain splash. In Coorg and Malland areas of India, slugs are found to carry the infective propagules of the pathogen. Management. So now we will discuss about management. So in management also we may divide it in cultural part, biological part, using resistant rootstocks, and using chemicals. So we will discuss all those things. So in cultural part, uh, in citrus orchards, we should go for improving the drainage to avoid water stagnation. We have to also avoid injuries which may occur due to use of implements during various farm operations. Orchards having thick weed growth or with green manuring crop provide favorable microclimatic condition of Phytophthora. So we have to remove unnecessary weeds also. Remove tender shoots near the ground level, which will minimize the incidence of Phytophthora stump rot. Periodical cleaning of moss helps to bring down the incidence of bark rotting. Post-monsoon earthing up after removing of dead bark up to a height of 6 to 8 inches of healthy portion of trunk is also found to be effective for managing citrus fruit rot as well as gummosis. For resistant root stocks, we can go for planting cultivars like Cleopatra mandarin. Cultivar Morocco is found to be resistant to Phytophthora palmivora. In biological method, it has been reported that biogens, mainly the fungal bioagent that is Trichoderma, having different types of species like Trichoderma harzianum, Trichoderma viditi, as well as another bioagent namely Glyocladium virens is found to be effective. Farmyard manure, poultry manure, coffee wastes, when they are added in the soil, it reduces the population of Phytophthora and increases the population of beneficial microorganisms. So for chemical method, we can go for use of Bordeaux paste in the ratio 4:4:40 or Chaubatia paste to control stump rot and gummosis. So these things we have to follow. We can go in a nutshell. We can say that we should use disease resistant rootstocks such as Rangpur lime or Cleopatra mandarin by keeping tree trunk free of new shoots or moss growth. We can also reduce the disease. Maintain drip circles well drained, free of weeds, or go for Green manuring during